Hello, everyone. Thank you for coming to my session. So today, I want to talk to you about 3 stories. So the first one is to introduce you to CSIRO, which is Australia's government research agency, if you're not familiar with it. The second one is using vanilla, so in this crowd I probably can say it, vanilla Spark, so a managed Spark instance. And the third one is around serverless architecture. So with this, let's jump straight into who CSIRO actually is. So CSIRO has 5,000 talented staff distributed all over the country. We have a very healthy 1 billion research budget. And our research stretches from material science all the way to astronomy. And I'm in the middle with health science. So probably the most famous invention that CSIRO produced, because we're famous, or we're passionate about translating research into products that people use on their everyday lives. So the most famous invention is Wi-Fi. But of course, we've done some other inventions. So for example, the polymer banknotes that you have in your pocket was invented by CSIRO as well and is now exported all over the world. UK is adopting it this year as well. And closer to my area, there's barley max, which is a grain that is higher in fiber and therefore is better for you and your gut health. So my research program is the eHealth research program the majority is in Brisbane. So therefore, our Wi-Fi equivalent product is called CardiHub. CardiHub was the first mobile health application that was used in a clinical trial, and it was for to help people do the rehabilitation after they suffered a cardiac event. So you would think that having a heart attack is a pretty good incentive for changing your lifestyle behaviors. Turns out it's not. Therefore, having a mobile app with a gamified user interface improved the uptake by 30% and the completion by 70%. So this little app has really saved lives already. So closer to my area, which is genomics, I don't know how much familiar you are with genomics, but let me say that all of our cells have one genome. So, or the other way around, the genome has given rise to all the tissues in our body. Therefore, all the diseases, the health risks, and even our behavior is influenced by our genome. So the examples that I have on the slides here is the hitchhiker's thumb, which is the last digit. Depending on your genes, it can go further to the back or not. So obviously, I don't have that gene. You can test yourself whether you have that particular gene. In the middle, of course, is disease risk. So the classic example here for a genetic disease is cystic fibrosis. And on the fun note, your genome influences the way you perceive taste. So for example, coriander, probably 2/3 of the people in the room really like coriander, which means 1/3 of you really hate it with a passion. Are there any coriander haters in the room? Well, I definitely don't like coriander, so that is, just blame the genome for it. So therefore, it's not surprising that the genome is, or can be used in the medical practice today. With that, the uptake of genome in the medical sphere has really skyrocketed. So it is predicted that by 2025, 50% of the world population have been sequenced, which is a staggering amount of data that is coming out our way. So this is a real data tsunami. And it is predicted that by the end of 2025, we have 20 exabytes produced every year in data, and this dwarfs astronomy, Twitter, and YouTube combined. So this is really something that, a data tsunami that is unprecedented, even in the commercial log file transformation, if you think of that space. And what is it actually used for? So this is a genomic research workflow that I used from the Project Mine, which is one of our collaborators. So Project Mine has the aim of identifying what are the genes that cause ALS, which is a disease that Stephen Hawking has, and you might be familiar with from the Ice Bucket Challenge. So therefore, the first step in this research workflow is to produce the actual data from the blood. And you will be hearing later today from Genome One a presentation of how this data is transformed into usable digital information. The second step is then to create profiles for each individual. The third step is then to combine this information and compare between individuals or build cohorts. The fourth step is to identify the disease genes, and then 5 and 6 is to find the underlying biological mechanism and find a cure respectively. So my team focuses on the middle bit, so combining information from large cohorts and finding the actual disease genes. So to walk you through the actual process, and mind you, this is a little bit simplified, in that we do identify the differences between individuals. So between you and the person next to you, there are sort of 2 million differences, because 99% of the genome is the same, but there are on average 2 million differences between you and the person next to you, which describes your unique physiology, but also your risk for disease. So therefore, in order to find the disease gene, you need to collect cohorts. So therefore, you have the healthy individuals as the control, and the people that are suffering from ALS as the cases. And the idea is that you want to identify the commonality between the cases that are different from the controls in order to identify the disease genes. And this is basically just a machine learning problem. So if you're familiar with the classical machine learning paradigm, you have your samples. On the y-axis, you have your features. In our case, these features are the genomic differences between individuals as your features in a matrix. And then you have the disease status, which is either healthy or diseased. And what you want to identify is the feature that is predictive of the disease status. Now, the only problem here is that we're dealing with a matrix that is 1.7 trillion, or that has 1.7 trillion data points. And this is really unprecedented in the machine learning space. So unfortunately, there was nothing we could take off the shelf to actually solve such a high-dimensional data problem. So we came up with a solution ourselves. So we came up with a solution ourselves, and this uses Spark, Hadoop Spark. So the way that I think about Hadoop Spark is going all the way back to your desktop compute. So on your desktop compute, you have your individual CPUs that are sitting there, and you might have a chip that has 10 CPUs on there. So therefore, the focus of desktop computes are real small data, instant access, personalized analysis on it. The next step then up in compute, in the compute landscape, if you want, is high-performance compute clusters. So with high-performance compute clusters, you do have multiple of these chips and with multiple of these nodes. So therefore, this will be in the orders of hundreds of CPUs, or in probably nowadays, it will be more closer to 1,000. But the point is that the focus of high-performance compute is compute-intensive tasks. So this will be stuff that, for example, the Bureau of Meteorology is doing, where they have individual tasks that can be solved without having to have data transferred between each other, between each of the nodes. So these could be independent solutions that you can parallel, or that you can calculate in parallel. But our use case here is that we want to analyze the whole cohort of information. Therefore, we need to have access to every data point of these 1.7 trillion available cells. Therefore, our use case is really data intensive. And data intensive, you not only want to have access to those CPUs independently, you want to join them together. So therefore, the way that I think of Spark and Hadoop is that it dissolves the boundary between nodes. Therefore, all the CPUs that are available in one Spark cluster, I think of as one entity where the communications between these CPUs and the nodes are standardized. So therefore, building on this framework, we developed Variant Spark. So Variant Spark is sitting on the Spark core, just like Spark ML or MLlib would, but it has the benefit of being able to deal with these high-dimensional data sets of millions of features. And therefore, what it can do is it can learn from 3,000 individuals, 80 million variants each in just 30 minutes. So this brings us really close to what is needed in the clinical practice. So the publication was from 2015, and it has now risen to the top 5% of all research output as measured by Almatric. And the reason for that is that it both improves on the accuracy as well as the speed compared to our competitors. So for example, MLlib, which is the commercial product that is, well, the product that is developed by the open source community, but for the standard commercial machine learning applications where you have lots of samples, but not that many data points per sample. SparkML was the solution that they developed for that. And it cannot deal with the high-dimensional data that we're dealing with here. So therefore, both speed and accuracy was inferior to what we produced. And therefore, Variant Spark is running on a MapReduce, well, an elastic MapReduce cluster, and therefore is basically available. And the way to make it even more available to our research collaborators is by partnering with Databricks. So if you're not familiar with Databricks is basically a notebook where you can write your code, you can share your code with other people. But the benefit here is that you can attach it to any Amazon resource instance that you want. So therefore, you can attach it to Amazon or the managed Spark clusters or MapReduce clusters. And you can also attach it to an S3 bucket. So with this, we therefore have the compute crunch of an S3, the access of an S3 bucket, but access through the compute power of a Spark cluster. And what I'm showing over here is basically the individuals. They can just link into this notebook and share the analysis. And in terms of research, this is exactly what global research is supposed to be, that researchers share their approaches, reproducible research, and therefore arrive at conclusions faster. So in order to demonstrate what you can actually do with this technology, we put together a demonstration case. Now, obviously, we couldn't use real medical data in that application case, but we came up with a toy data set, if you want, or a synthetic data set. So therefore, we chose 4 positions in the genome, which we treat as a disease genes. And in this formula that I'm showing down here, we put the content of these genomic loci together in this highly complicated formula, which represents how complicated diseases actually work together. So therefore, the effects from the genome might be additive, they might be multiplicative, and they certainly work together. So therefore, with this, we basically sum up or multiply the content of each of those genomic locations to produce a score for each individual. And depending if that score is a certain threshold or below a certain threshold, we annotate it with either disease or non-disease. So in our particular case, this synthetic data set was the Hipster Index. And those 4 locations that we used were reflective of whether someone has a monobrow or is likely to have a monobrow, what their facial hair status is like, whether they, receptors in their eyes can detect textures better than others, and similarly with the coffee consumption. So therefore, remember, what we've done is we put this through the formula, and at the end only had a 1 or a 0 for each individual. And then the full genomic profile with the 1 and 0 location, we put that into Variant Spark in order to rediscover if you want, rediscover these original disease genes. And as you can see from the picture down here, in gray I'm showing all the genomic background, which is the noise. And then in color is highlighted the individual disease genes that were truly used in order to build that phenotype. So therefore, with this, it is clear that Variant Spark can pick up a complex, multivariate disease, if you want. Here, in our case, the Hipster Index. And it will be a similar thing for complex diseases. And you can use that data, or you can use that methodology on your own data, because obviously this is not limited to genomic data. Every discipline that has high-dimensional data sets and a machine learning problem they want to solve can use this technology. So therefore, I encourage you to go to Databricks, where you can point Variant Spark to your S3 bucket, to your data, and identify your feature that is predictive of the variant. Okay, so with that, I do want to change track and talk about DTScan2, which is a serverless architecture for genome engineering. So, if you're not familiar with genome engineering is a new discipline, molecular discipline, that really has revolutionizing property. Specifically, it has the potential of curing genetic diseases. And with that, it can ring in a golden age of medicine, where cystic fibrosis or hypertrophic cardiomyopathy are things of the past. So specifically for hypertrophic cardiomyopathy, that's a disease 1 in 500 Australians actually carry, and the worst case scenario is that you drop dead from a cardiac arrest because your heart walls have thickened or have enlarged. And the worst part about that is that you might not even be aware of it. So a lot of these people that are actually carrying this, predisposed to this disease, are not aware of that. So therefore, if you can just go in and edit that beforehand, before the babies grow up and actually have that disease, that would be good. And how close we actually are to actually doing that was demonstrated by a Nature paper which came out probably 2 months ago, where they went in and actually edited human embryos to be free of this disease. The downside, though, is that it did not work every time. It only worked in 7 out of 10 embryos, which means that 3 out of 10, this technology failed. And if you think of this being your unborn child, then 3 out of 10 is just unacceptably high. So this is where DTScan2 comes in. DTScan2 has the aim of making this technology work the first time, every time. So therefore, the first thing that you need to solve there is how to actually target a gene. Like, what is the address of a gene? A cell does not have street or house numbers. What we need to find, though, is a sequence close to the gene that is unique enough for the rest of the genome, for the editing mechanism to bind. And since the genome is built of 4 letters in a string of 3 billion length, sequences can occur randomly. And this is sort of represented here by houses that just look identical. Therefore, how would you target a house that looks identical? And with that, the task is basically to search the whole genome for something that is absolutely unique. That is something that the mechanism can target, but only occurs in the genome once. And this is a highly computationally expensive task. So, and the added issue to that is that people might not want to search one gene. They might want to search 100,000 genes, so all the genes that are in the genome. And in order to do that, has only recently become possible. Because these individual tasks, or these individual searches, they're quite quick. They can be in the order of a couple of seconds. But then scaling that up to 100,000 genes is not trivial in the cloud. So serverless architecture came in, and that was basically match made in heaven, because it allows us to scale up tasks that are as agile as what we need. So therefore, the way I think about serverless in terms of the focus is agility. So here, it might be data intensive. You can string together a lot of Lambda functions and achieve data intensive compute, but the agility is key. So therefore, the way I conceptualize serverless CPUs is that they are basically free-floating CPUs that you can recruit instantaneously without the lag time of spinning up a new instance. The problem, though, is that Lambda was designed for individual quick tasks, like speech recognition in the presentation that we heard before. Whereas genome analysis is a bit more complicated than just translating an audio track into letters. In fact, the genome is quite large, and the memory constraints or the storage constraints on Lambda just do not fit. Therefore, we had to come up with a workaround to spawn out many Lambda functions and collect them back. We were quite fortunate that the genome comes in natural partitions already, called chromosomes. Therefore, we came up with a way of spawning out search functions for each chromosome independently and then collecting back the information together to display to the user. Because ultimately, what GTScan2 is a search function or search engine for the genome, where it comes up with, or it ranks all the options of how to target a gene, so researchers can pick the top one, and hopefully this is the one that worked the first time, every time. So therefore, the architecture that we came up with is that we have a search, which is basically a regular expression going through the genome to identify all the possible targets. It then triggers 3 separate Lambda functions, which evaluates the properties of how to target those individual calls, which then feed into a 3rd Lambda function, and ultimately all of this goes back to the user in real time. So therefore, the user interface will be populated while the search is going on in the background. So this is a more Amazon-y architecture. And if you want to know more about this, we just recorded a This Is My Architecture series, which should be released within the next couple of months. So with that, GTScan2 really has the ability of bringing the latest in CSIRO scientific excellency together with the latest in the compute world in order to bring a solution to researchers that really can solve a tricky problem of how to target or how to make this technology work the first time, every time. As such, it was highlighted, or it got a lot of attention by the international IT community, with being featured on AWS blogs, at the CIO magazine and Genome Web, and we also are currently in the running for winning an I award in Melbourne. And basically, what I'm just showing here with this slide is that we improved the accuracy already by 30%, and we reduced the runtime from actual weeks to seconds. Therefore, it has a very phenomenal uptake of 3,600 function calls already on the technology. And GTScan2 is freely available should you be a genomic engineer. So it's freely available at that URL. So with this, I want to summarize how AWS technology has actually helped us achieve research excellency in that space. So therefore, it really has improved the time to science. And by that, I mean that we don't have to manage clusters. We don't have to patch anything. And our users do not have to install the actual program. So the way usually bioinformatics works is that you develop your beautiful algorithm, you stick it into a Git repository or ship it as a binary, and people then have to figure out how to use it on their own infrastructure. So I'm sure it's the same thing in the commercial world. And that is a huge barrier, obviously, because your environment is not the environment that they envision, and it's just a horrible situation. So therefore, being able to deploy something with serverless architecture where it's just in the cloud and does not rack up a huge instance bill, because it only uses as much, it only incurs cost for the time the Lambda function is actually running. That has really made a huge difference in our workflow. Similarly, that we can spin up clusters on demand. So traditionally, we have our high-performance compute cluster, but there, everyone else in the organization wants to have access to. So you're stuck in a queue, and at the end, you reduce down your number of CPUs just to get it running, which is not, what a high-performance compute cluster is about. Therefore, spinning something up that is appropriately scaled for the question that we have at hand makes a huge difference. Also, the ability to share information with our international collaborators, because there are obviously different data centers all over the world, is again a huge benefit. In the past, we had to log into a cloud or a cluster that is in Europe, and therefore the lag time of just typing in ls, enter, and it took forever to actually come up with the results. That kills research enthusiasm and research workflows. Therefore, now we can just deploy a solution. We can test it on our own region, our own data center here, and then deploy code that is working for sure in other data centers for there to run. The second part is that it is improving outcomes for research dollars. So by that, I mean that all the money that has gone previously into building infrastructure now goes into running the actual research tasks. And the other big thing is that we've been developing Spark for the last couple of solutions on top of Spark for the last couple of years. But it's only been recently that we actually had access to a Spark cluster. So given this lag in how quickly a technology is actually introduced into the community, when it has to be a physical thing that you buy, is just not acceptable, because it's accelerating so fast that you do want to play with the latest, newest, shiniest toy right now, rather than in 3 years' time where it's outdated already. Therefore, renting something from Amazon where we don't have to put in physically in our data or in our IT center is a real privilege, I have to say. Good. With that, I want to conclude by thanking the team. So the other thing that I should point out here is that you can see it's very male-dominated, my team. Therefore, the influencers here in the room, we do need to work on the pipeline of bringing more females into the field, of really having a healthy pipeline that is gender-balanced in order for us to then at the end hire that. I encourage you to all think about that and work on building a healthy pipeline so we can have more diverse teams and ultimately better outcomes in the future. With that, thank you very much.